Professor Pati works in the field of plant biotechnology with a major focus on medicinal plant biotechnology and has more than 80 international publications. Wow, sir. He has 3 patents and 1 process that is commercialized. Professor Pati has handled several research grants with around 3 crore rupees from different funding agencies like DBT, DST, CSIR, UGC, DRDO, etc. Professor Pati has visited 11 countries, including Japan, USA, France, Italy, Philippines, Norway, Belgium, and Israel. Professor Pati is the recipient of various prestigious awards like Boy Scout Fellowship of Department of Science and Technology, Government of India, Fulbright Nehru Senior Research Fellowship, Generation Challenge Award, and Commonwealth Travel Grants, etc. He has also been conferred INSA Teachers Award from Indian National Academy of Sciences, New Delhi, which was instituted to value constituent and high level of teaching in educational institutes. Professor Pati is also recognized as an outstanding performer in Biotech Career Ready Reconner 2014, published by VV5 Media Private Limited in association with DBT, Government of India. As a mentor, his students have brought laurels to the country by receiving many prestigious international fellowships such as Fulbright, Nehru Doctoral Fellowship, AIST Japan Fellowship, BELSPO Fellowship, etc. 2 of Sir's PhD students have also received Young Scientist Awards, and one at present working with Nobel laureate Dr. Christian Walhard at Max Planck Institute, Germany. Professor Pati is a member of more than 20 different committees of Guru Nanak Dev University. He has served as a coordinator to organize global initiative of academic network GYAN of Ministry of HRD, Fulbright Specialist Program of USIEF USIF, and many national and international conferences. Sir is also a NAC assessor, NAAC NAC, and member of several national committees, including Reviewer of Fulbright Doctoral and Postdoctoral Fellowship, DBT JRF Program Committee for Preparation of Syllabus and Policy Framing, and a member task force for Star College Scheme of Department of Biotechnology, Government of India, New Delhi. With this brief introduction, again, I would like to invite Professor Pati. Please take over the charge now, and we are waiting for your talk. We'll enjoy, sir, definitely. Thank you, sir.
Thank you. If you allow me to share the screen, please.
Mafatullah, do we have, sir?
Who's disabled the participant?
Hello, you have to share your screen, sir.
So when I click on it,
click on share screen, sir.
It says who's disabled. Screen share.
Okay, I will ask our IT team, sir. Let me ask.
With sir's presentation we have, Mafatullah.
Sir, I don't have his presentation. I will ask IT team to enable.
I can do it.
Right.
Are you able to see the screen now?
No, till now not, sir.
Sir, please check.
Check now, sir.
Are you able to see?
Yes, sir. Yes, sir. Yes, sir. Yes, sir. Yeah, fantastic, sir. Please.
Thank you, Dr. Shekhawat. Good evening to all participants and the resource person. And in fact, it's a treat to listen to Dr. Professor Rajan, and he has been a teacher, and it's a pleasure to have him in the same platform. But equally, it is difficult to speak after him. I mean, we have grown reading his research papers and his books, so it's always difficult. So my regards to sir for sharing the same platform. And I also convey my regards to other resource persons who joined webinar. Fantastic beginning. Credit goes to Dr. Kher, Dr. Sekhawat, and all the team who were behind this. So it's great to know that such a large participant they registered for this webinar. So all credit goes to the organizer. In fact, my area of specialization is medicinal plant biotechnology. Since long, I've been working on this area, and as the theme of this webinar is concerned, I thought I should make my presentation as student-centric, give a general lecture to students, and give them a kind of team where they could really pursue their future research. So I thought I should speak on hormonal biotechnology, and rightly so, because if you see the new Millennium Goal of the United Nations, the priority is health and nutrition, apart from education, environment, and other things. So I thought I should talk about how technological intervention can play a very important role to prepare plants for good nutrition and for better health. So that is what is my theme of today's lecture. So when we talk about plant, you all know that it's said that God slips in mineral, awakens in plant, works in animal, and thinks in man. So we always see plants in the form of God. And if you look at our ancient literature, we always treat plant in the form of a deity, and we worship, we revere plants, and plants are a kind of let's say it is in our daily life, it is in our rituals and all. So plants is inestimably linked to us. And if you look at the application of plants in modern days, we know the application of plant, but in ancient times also, the application of the plants is very well documented, whether it is in the field of textile industry or it in the field of cosmetic industry or food fibers. So their application is very well documented. And as far as the application of these plants in medicine, that is also in our ancient scripture. It is very well depicted. And when we talk about the use of these plants in the form of medicine, then it's very difficult to draw a historical perspective. I mean, nobody knows when we started realizing that the plants can be used in the form of medicine. But probably it is an accidental discovery that when people started using plant in the form of food and it has subsided the pain and they've started realizing probably the plant could be a source of medicine. And if you look at some of these ancient literature, whether it is Sumerian clay or whether it is Egyptian civilization or for that matter Chinese literature, you find it's a very old science. It's a pretty old, maybe 3,000, 4,000 year old science. And even if you see our Indian literature also, and it gives us a kind of feeling that it is probably thousands of years old, particularly it dates back to Rig Veda, which is several thousand years old. So that's the kind of history that goes with these medicinal plant or herbal plants. Okay, so when we talk about herbs, naturally Ayurveda comes to our mind. And we all know that when we talk about Ayurveda, this Ayurveda is derived from two words. Ayu means it's a life, and Veda means is the knowledge of science. And we call Ayurveda as the science of life. And it not only is used to heal diseases, but it is a complete way of life. It gives us a happy, healthy, and peaceful society. And it serves two purposes. One is it provides health to healthy people, and it cures diseases to sick people. So that is the two different purposes that it solves, and that's why this science has been practiced since long. And recently there has been a resurgence of interest in this herbal medicine or plant-based medicine, which we call as phytomedicine or botanical medicine, because we find that there has been a rising cost of medical care. And we also find there are several side effects of allopathic or chemical drugs. And we also see that these herbal drugs, they are easily available. You can fetch it from natural sources, and you don't require an expert to be consulted for this matter. Maybe we sometimes we call it as grandmother's recipe. So we all know the uses of this plant, and they can be easily used. And it goes to the root cause of the problem, and it addresses some of the obstinate diseases. And that's why this has been so popularly used all over the world. And the Indians were very proud because it's an old science and we use it very frequently. If you look at the application of these herbal medicine or plant-based medicine, you'll be happy to know that around 80% of this world population, particularly in developing countries, still use this as a primary source of medicine or healthcare. And if you look at some of the statistics, you will find that 1/4 of the prescribed drug either contain plant extract or contain some ingredients derived from plants and or plant substances. So that's the kind of situation when we talk about herbal medicine. And if you look at the economic aspect, very encouraging. Plant-based medicine at this point of time, the global market is 62 billion US dollar, and in India it is 1 billion US dollar. And you see this annual growth of 15%. Every year you find this jump, and it accounts for 30 to 40% of total healthcare expenditure. Huge market. And if you look at the projections, very encouraging. And you will see that this resurgence in herbal, the interest in herbal products, which is now at present, it is 120 US billion dollar. It is projected to reach around 7 trillion by 2050. That's the kind of projection. This is very, very encouraging. So now if you go to the market, you find the range of these herbal products, flooded with herbal products. And these products are booming. And there is a huge potential of these herbal products because people are more aware of using natural products and all. And these natural products are derived from different parts of the plant. You will see that leaves, rhizomes, roots, flowers, seeds, all these different parts of the plant is used in herbal industry because these parts probably contain the active ingredient which is used for having therapeutic potential. We look at the plant species and the medicinal plant species country-wise. You will find India and China is at the top. They have a lot of these medicinal plants, and in fact, if you look at the research paper that is coming from these two countries, they are fairly doing well. So a lot of publications, medicinal plants, they are coming from India and China. And if you compare all these different kind of complementary health system of medicines, whether it is deep breathing, whether it is homeopathy, whether it is whether it is yoga and all, you find natural products is highest, it is top in the list. Around 17 to 18% of this is being used for healthcare, and that's why there has been a lot of scope to work in this area. And you will find there are many medicinal plant. Professor Radhan also pointed out some taxons and all. You will find Podophyllum, you will find Papaver. There are many medicinal plants which we know their active ingredient, which we know their therapeutic potential, and people have started after clinical research. They have started synthesizing some of the synthetic and semi-synthetic drugs out of that. And there are many plants which are in the preclinical stage, and where people are exploring every day new molecules and all, which is used for development of plant-based medicine. And if you look at the secondary metabolite, which I told you that these plants, they contain different secondary metabolites which are pharmaceutically important, and there are huge range of secondary metabolites which are present in the plant. And these secondary metabolites are mainly derived from primary metabolites, and these primary metabolites, for example, your cyanogenic glucosides or cardiac glucosides or alkaloids, phenols, propanoids, the major class of secondary metabolites. They are derived from primary metabolite. For example, glucosides, they are derived from sugars. And similarly, alkaloids, they are derived from amino acids. And topinoids, they are derived from respiratory pathways and all. So you find wide range of these metabolites synthesized from primary metabolites. The other aspect which bother us is the challenges. The challenges in the medicinal plant. But when we work on medicinal plant, we need to be aware of the challenges. And the major challenges in medicinal plant is the greatest irony is when once we know the potential of these medicinal plants, we go and harvest this medicinal plant from the natural habitat. So that is a indiscriminate harvesting of this medicinal plant. As a result of this, their population is dwindling, and we need to seriously think that how we can address this challenge. Then there are many cases where we find that the agrotechnology is not very well developed. We do not have propagation methods of medicinal plant. Then the quality is a big issue, and that's why a lot of R&D is required to address these challenges. And in fact, if we can work on these challenges, probably this herbal medicine will become a safe, effective, and affordable system of healthcare. So these are the challenges, and here comes the technological intervention. So looking at the challenges, we can think how technology can really be used to address some of these challenges. So my second part of the talk will be how technology can really help, or technological intervention can really help to address some of these challenges. So when we talk about technology, then probably 21st century is known the century of biology, and biological, particularly biotechnological intervention could be really help in addressing some of these challenges. So the first challenge is when we talk about health and nutrition and then there's the plant. So the value addition of the crop, how we can add value to the crop. So we will touch upon some of the technology which is available now. Then how we can grow scientifically the plant, to propagate these plants scientifically. The theme of this webinar is on scientific propagation, use of technology, particularly agriculture and all, and protective cultivations, how we can grow these plants. Then quality is a very important aspect. Then improvement of the plant is, of course, very, very important. So I'll give you some examples how people are working in this area, and how technology is helping in these areas. So when we talk about functional food, basically we take food because of its nutritional value. But when we talk about functional food, we see beyond its nutritional value, the physiological health benefit is in the top priority. And in fact, there is a kind of a paradigm shift these days. So we consider that let food be the medicine and medicine be the food. So this has already changed our narrative. Don't just survive, you have to thrive. And I am reminded of the saying of Royal Lee, who is considered as the father of holistic nutrition, in one of his books, Therapeutic Manual. And I would like to underline this statement, and this is very, very important. One of the biggest tragedy of human civilization is the precedence of chemical therapy over nutrition. It's a substitution of artificial therapy over nutrition, over nature, poison over food, and we are feeding people poisons trying to correct the reaction of starvation. So here, it's a very symbolic statement. We have been taking medicines, but probably we may not have to take medicine. Maybe corrected if you take right kind of food, right kind of nutrition, and change our lifestyle. Probably that's going to help us. But mostly we take chemicals, and which really have adverse effect to our human health. So how can we make a food a functional food? Probably there are different dimensions which need to be thought over. Probably we have to add a component normally which is not available to a plant. So how can we add that component? And then maybe we can increase a specific component. Maybe a phytochemical we can increase, and we can increase the concentration of the bioactive compound, and that might help us. And then we can replace, because there are crops where you find one of these phytochemicals which is not much warranted, and you want to replace it. How we can replace that component? And then bioavailability is a big issue. Many researchers are working on bioavailability. For example, curcumin. Curcumin is a very important pharmaceutical important component which is found in turmeric. But how we can increase the bioavailability of curcumin? Maybe you will see these days it's coming to market that some people they are black paper to it, so that bioavailability of curcumin increases. So how biotechnology is addressing these issues? So in our first day, we discussed about tissue culture, genetic engineering. I just give an example. I'll quickly go through this, where people have been trying to add a specific component, because that's not there in that plant, but how they are adding it so that it is of more health benefit. Vitamin A is a very, very important component, and in fact, there are many health issues which are related to the deficiency of vitamin A. And you find in regions like India and many of these Asiatic countries, this is a big problem. So if you look at rice, the rice, if you look at the vitamin A biosynthetic pathway, you find there are many genes which are absent. For example, phytotin synthase, phytotin desaturase, beta-carotene desaturase, lycopene. These are absent in rice, and hence rice cannot synthesize vitamin A. And in fact, in underdeveloped or developing countries, rice is a major source of food security. So how can we incorporate this so that we add value to rice? So that's why you must have heard about golden rice, where people have tried to increase phytotin synthase, adding this gene from daffodil. Then you have phytotin desaturase and beta-carotene. This is added from bacterial sources. Again, lycopene from daffodil. So whole biosynthetic pathway of synthesis of vitamin E is complete, and that's how it has given us the golden rice, which is increased with vitamin A. So this is the first example where you can do pyramiding of genes, and you can increase or you can add the value to this crop, because earlier vitamin A is not synthesized in rice, but now it can be synthesized or it can be biosynthesized in rice. which will help millions of people who are deficient of vitamin A. There's another example. This plant is known as Ricardella dulcibeca. This is a plant it grows in West Africa, and it bears a kind of fruit which is red in color. And this fruit it synthesizes a protein, and this protein is tasteless, and but it has a unusual property. When it comes with a sour taste fruit, it makes it sweet to taste, and that's why this kind of property is being now added to crop for example, tomato, grapes, maybe citrus fruit, where it can make it sweet and it can add value to that. So this is a kind of addition of a trait where it doesn't exist at all. Look at this kind of cauliflower. You might have seen cauliflower, which is white in color. These are white florets. But this is not just to add a splash to your dining table, but these are genuine scientific experiments, breeding experiment, where people have added pigments which have great nutritional value, which has great health value. These are anthocyanin, carotenes, and all. They can protect you from cardiovascular diseases. They can protect you from cancer and many other. You adding this where it is not present earlier. Similarly, examples of tomato. Now you can see in market you can find tomato which is of different color. So the anthocyanin, there are different approaches to which this tomato are produced. Maybe some breeding experiment, maybe with some addition of genes, incorporation of genes. So you can get these kind of tomato which are anthocyanin rich, and they are better for your health and can protect you from a lot of diseases. Similarly, addition of value to broccoli, squash, which is you can increase them with some antioxidant, glucosinolates, and all, and they have more commercial value. So these are things which you can do. This is just enhancing the particular phytochemicals. In earlier I said how can add a specific component. Here you can enhance the value of a specific component. Then there came technology, which is known as gene silencing technology. People got Nobel Prize for this technology, which is known as the RNA interference technology. And these technology also added new dimension to crop improvement, and where you can produce crop which are novel, which can nutritionally rich, and which are disease free. And this works on the principle that if you all understand the central dogma of molecular biology, we see the flow of information from DNA to mRNA to protein. And if you can block the information at the mRNA level, you don't get the protein, and protein usually control the trait. And that is what is the post-transcriptional gene silencing is known as RNAi or RNA interference technology. So how this technology is helping to produce the crops which are nutritionally rich, or you can amend some of the crops which are better for your health. I give you one example which came in Proceeding of National Academy of Sciences. This is about gossipal story, where gossipal, you all know that in cottonseed this is present. This is a toxic topinoids, and this in fact has a lot of adverse effect. It can lead to kidney malfunction, and in extreme cases that can cause paralysis. So scientists, they have identified that the gene which helps in production of this gossipal, delta cadherin synthase, is a key rate limiting gene in the gossipal production, and this has been silenced using RNA interference technology. And as a result of that, you get a crop which is very suitable for consumption of cattle, and humans, pigs, and all. So that's how the technology is moving. The other example is the cassava. In cassava, you find cassava is a very, very important tropical plant. In fact, it ranks at the fourth in production of all tropical plants. Huge role in food security. But if you look at the problem of cassava, it produces a cyanogenic glucosides known as liramine. And when you consume this, it goes to your intestine. It acts by enzymes and the flora of intestine, and it converts it to notostaline and decomposes it to toxic chemicals like hydrogen cyanide, which is not good for our health. So people have identified how liramine is produced. You could identify there are two seeds which are C450, which are essential for production of liramine, and they have tried to silence this two gene, and as a result of this, liramine production has been avoided, and that's how you engineer plants for better health. An example of low gluten rice, because you will see that most of the kidney patients they are recommended to have low protein diet. So rice is consumed by many people across the world. So how to produce a rice which is of low protein content? So scientists could identify one gene which is silenced, that is called gluten B. If it is silenced, then probably we can produce a rice which is of low gluten content. And that is how, in using RNA interference technology, this gene was silenced, and in normal rice you could produce a low gluten rice. So that is what it was published in Plant Cell in 2003. You can read the detailed story. The reference are given just below my slide. So the example of coffee, how you have improved coffee. So I was talking to you that some of the toxic compounds can be removed by technology. So in coffee, it produces great number of alkaloids and all. Some of these alkaloids are good for you. Some of these alkaloids are not good for you. For example, caffeine. Caffeine is an alkaloid which, if you see, a cup of coffee contains around 158 mg of caffeine, and the caffeine it is used for insomnia, restlessness, palpitation. They are all adverse effects. And so scientists thought how we can remove this from coffee so that we make it more appreciated by people and with less health adverse effect. And the chemistry people that chip in, and they said that we can do it with supercritical extraction. We can remove this caffeine out of it. But what happened is, once you do this, you lose the aroma of the coffee, you lose the taste of the coffee. And many people, they drink coffee because of its taste, because of its aroma. So then this technology of RNA interference technology was used, and the rate limiting enzyme for production of caffeine was identified. It was silenced, and as a result of this, you have reduced caffeine content in coffee, and it makes it more sense because it has more health benefit, because the caffeine has adverse effect to our health. Similarly, you might have heard about Webera cotton. This is an example I was giving you, that how we can make a food, a functional food, by more bioavailability. So in this maize, you see this is a very preferred crop because a lot of protein content, and people like it. But if you look at the essential amino acid, it is deficient of most of essential amino acids like lysine, like tryptophan, like methionine. It is deficient of all these. So how to make this maize a better crop? So if you understand why lysine is not available, you will understand that it is conjugates with beta-glutarate, and there are two enzymes. One is known as lysine beta-glutarate reductase, and the other enzyme is saccharophile dehydrogenase. They help in the conjugation of this lysine with beta-glutarate, and as a result of this, we don't get the free form of lysine. And that's why the scientists, they thought probably these two enzymes which helps in the conjugation may be silenced, and as a result of that, we can get the free form of lysine, which is essential amino acid, and that is the kind of variety which is produced in corn, which is known as a Lavera corn. Then came the challenge is how to grow, because we think that whenever there is an important medicinal plant or a plant which is of high value, it has been indiscriminately and we lose these important plants. And so scientific propagation methods should be in place for most of these important plants. And here comes the role of plant tissue culture. And you have been listening in some, since last 2 days about all these intricacies involved in plant tissue culture. I will not go into that. But you need to understand that this is a technology where a part of the plant, even a single cell, can produce the whole plant. Professor Radhan told you how protoplasts can produce a plant. It's a more kind of flexible approach where not even a single cell, even a protoplast can give you a plant. So it gives you tremendous flexibility of maneuvering. You can maneuver different traits when you have these technologies available. So you will see that there are many plants, medicinal plants, that are propagated in time. And lab is actively involved in standardizing, developing protocols for many of these plants. So we have developed protocols for cost-effective method of propagation of catharanthus. This catharanthus is known for its alkaloids like vincristine, vinblastine, which is used for blood cancer. So in our laboratory, we try to increase these bioactive compounds using plant tissue culture. Similarly, we have been working in Vidhania for long, and we have developed protocols for its regeneration, for its pathway modulations, and this plant is very, very important for several diseases, whether you name it as cancer or an immunomodulator for viability. It has a tremendous role. And in fact, in Amritsar itself, it is a 1,000 crore business of single plant. So that's the kind of potential of this plant. So we have developed a very robust system for its propagation. Back to back, there are 2-3 couple of papers are coming now on its regeneration protocols, on its micropropagation protocol. Recently, one has come in industrial crop, and one is shortly coming in industrial crop. So that's kind of technology that need to be developed. So if you have a elite plant, you can propagate them very fast. Similarly, you can look at picoryza. Picoryza is a very important plant because in all liver formulation, you see this is used. This is known as putty. So the rhizome, the root has a lot of medicinal value. So how can multiply these plants so that it will be of great value to our herbal industry? Tylophora, we have recently published a paper on Tylophora. This is a very important medicinal plant, particularly in asthma. It has a lot of use, and we developed a protocol, and we did some preclinical studies on Tylophora. Stevia, you all might be knowing about this plant. That's a very important plant, particularly because of its stevosides. The stevoside is a sweetener, and those who are diabetic patients, they cannot take a normal sugar. This is used as a sweetener. They have very important role against diabetic ulcer. The other plant is very close to my chest. This is rhodiola. And rhodiola is, you all might be aware that when our Prime Minister was addressing the nation on Article 370, he mentioned about this plant, the solar plant. And this plant, it grows in high altitude. In the Himalayan region, there's a snow line. And this plant has tremendous value because when our military personnel, they go to high altitude, there is a hypoxic conditions. And under this hypoxic condition, it is very difficult to survive. And once people they know the importance of this plant, they have there has been depletion of this plant. And now, if you go there, you find very few plants there present. So the Defense Organization wanted that we should multiply these plants in our laboratory conditions and a proof of concept and put them back to the nature. And we have been very successful in doing that. And we have grown this, we have developed this protocol, and now we are mass propagating this plant in our laboratory. So what I mean to say is medicinal plants are very, very important. And now the kind of technology that is coming in this, we need to have a collection center, and we need to have a multiplication facility. So tissue culture can be used in multiplication, and then we need to look at this kind of arrangement in Sri Lanka, where there is a Nature's Beauty Creations, where they have collected so many medicinal plants, and there is a kind of facility where apply these plants and can go back to industry, and entrepreneurs can really look into such kind of commercial business where because the scope of medicinal plant is huge. In the beginning I told you how it is picking up, and this is a kind of technology where can multiply this plant. Not only tissue culture, now hydroponics, aeroponics systems are coming in growing these humble plants. And this is one of our collaborators. They have developed a kind of hydroponics facility in Vidhania. We work on Vidhania, so we thought why not develop a kind of technology for growing them under hydroponic system. So look at this, how they are growing luxuriantly. Look at the root system, the luxuriant root system. So this is kind of opening that one can think of. Similarly, look at this artemisia growing in under a hydroponic system. You all know that artemisinin is a very important ingredient, important compound which is present in artemisia. And you got Nobel Prize in 2015 for discovering this artemisinin. This is an anti-malarial drug, and this can be grown because it has a lot of commercial value. So it can be grown under these kind of technology. Then plant tissue culture plant cell culture could also be additional technology which can help herbal industry. I will give you a couple of examples. So you grow callus, you grow cell suspension, and then you bulk up these cell suspension. Dr. Professor Razdan was giving an example of ciconine from Lithospermum erythrozoite. And there are many other plants also, they can produce ciconine, like Arnibia can produce ciconine. So look at this, how a bioreactor is producing ciconine. And this red dye can be used as a biolipstick because the natural compound are more preferred than the synthetic compound. Synthetic compound might have adverse effect to your skin and all. So people are trying to develop these natural this biolipstick and food additives, dyes and all using these plant-based cell culture system. These are huge market. If you look at the commercial market of these metabolites which are derived from plant cells, this is not a story. It is in fact a reality. You can see now plant-based cell suspension culture-based metabolites are available in commercial scale for healthcare. Then the last slide, which I would like to show you to give an idea that, as I told you, that we have been working in the area of a medicinal plant. In medicinal plant, crop improvement is a very, very important aspect. So if you identify complementary traits where you need to improve, put them together and develop a variety which is acceptable and which is, can be considered an elite. So you have plants which produces a lot of metabolites. Then you have plants which are having a larger biomass, but they are sensitive to diseases. They produces less metabolite. Then you can bring them together and produce a good variety. You can improve them, but because genetic manipulation sometimes is not acceptable by public, in that case you can go for some kind of breeding where you can produce the novel variety. So we breed this different variety of Rasagandha and we produce Rasagandha in our laboratory through breeding. This is very elite and it has a lot of secondary metabolites, it has high vidhanol and it has resistant to diseases. It is also rich in antioxidant. You will find chlorogenic acid, fatty acid. There are many antioxidants which is very rich in this plant, and it has a vigorous growth. You can see the growth in a single plant. So you will find other attributes are very good, and it has a good anti-proliferative activities also. And we have this mass propagation protocol in place to multiply these and give it to farmers or on further. So we have been working how we can help these entrepreneurs and farmers, giving them a medicinal plant which can be grow and their income can be increased. So in my concluding statement, I would like to, because there are many students who have joined, so there is a trend which we see, it's back to nature. So we need to now care our nature. We have to really see how we can help and grow this plant in following a scientific rationale, and that will be a great service to the humanity and to the society. So once again, I thank all the organizers for giving me this opportunity, and for all the participants, I would request them eat good food and stay healthy. Thank you all.
Thank you very much, Professor Pratap Pati sir. We enjoyed your lecture. It's really colorful, sir. Like colorful fruits or food is available on the table.
Thank you.
Enjoyed, sir. There are some questions, sir. We can have these. One question from Bhumi Patel. Asking that Momodica chantrantia help the diabetes patients if they use in their daily diet. I think you understood that Momodica chantrantia.
Yes.
So is it helping in diabetes? That is the question, sir.
Yes, I think for the youngster, for the students, I'd like to tell two things to them. Number one is knowing about the use of this medicinal plant. That's one, right? So there are many
Yes, sir.
and we have traditional knowledge. We know about the function of the plant. Like the people say, Karela is good for diabetes. There are many other there are species of Vidania like Vidania coagulans. The fruit of this plant
Yes,
is
sir.
diabetic. So there are many such plants which are available, which are traditional knowledge. But what is required now? I'm coming to the second point. That is the challenge for our youngster. Is it has to be backed by a scientific evidence. That is where we require the help of these youngsters. They should work on this and find how we can support this, our traditional knowledge, backed by scientific evidence. So that is what is required, and if that can be done, because we do not know what is the dose to be taken, to what extent it has to be taken, because there are many secondary metabolites we take to a large extent, it might affect your health also. So these are the things which need to be done, and that is where it is very important. Knowing traditional knowledge is fine. That is narrow down our whole exercise and reach it somewhere, and after that you have to take it to the next level.
Yes, sir. Thank you, sir. So let me summarize sir's presentation about the herbal biotechnology. We learned about the golden rice, sir, and the problem with the beta carotene pathways. That miraculin, the taste modifying protein, sir. Rainbow cauliflower. It's the first time we enjoyed that, sir.
Now these are available in supermarket.
Oh, right. That name rainbow cauliflower is really fantastic. Anthocyanin containing pigment containing tomato, purple tomato, the RNAi technology, cassava, the lina marine chemical compound, low gluten rice, decaffeinated coffee, corn with lysine, low corn with the lysine, and finally, sir, concluded with the back to nature. That's the biggest message, sir, to the young generation. Thank you very much, sir, for the wonderful presentation. We enjoyed. Hope in future also we will enjoy your lecture, sir.
Thank you.
Thank you.
An opportunity. Thank you so much.
Thank you, sir. Now it's time to move on to the next talk. Good evening, sir. Dr. Sadanand, sir.
Hey, good evening, everyone.
Sir, good evening, sir. Welcome back, sir. So, with sir's brief introduction, we will go ahead with the talk, sir. Dr. Sadanandan Dekne is an associate professor of genetics and plant breeding at the University of Maryland, Eastern Shore. He received his PhD in horticulture from the University of Florida, which was followed by postdoctoral research at University of Florida, and assistant and associate professor's position at the University of Wyoming. Dr. Degne's research focuses on genetic improvement of perennial fruit species using conventional and molecular breeding approaches. His current research is directed towards grapevine genetic improvement using precision breeding and genome editing. Dr. Degne received the 2013 Young Scientist Award from the Society for In Vitro Biology. Wonderful, sir. The 2016 Early Career Research Achievement Award from the University of Wyoming, and the 2017 Excellence in Research and Teaching Award from the National Agricultural Honor Society. He has published more than 50 peer-reviewed research articles, in addition to giving several talks at national and international conferences. With this brief introduction, sir, I invite you for the talk, sir. We are waiting for your talk, sir. Thank you, sir.
All right. Thank you, Dr. Shekhawat. I'm just going to try to share my screen, and let's see if I can do that. All right. Can you guys see my screen?
Yeah, it is.
Okay, awesome. All right. Good evening, everyone. It's
yeah.
Can you hear me?
Yeah, fantastic, sir.
Yeah. Good evening, everyone. It's around 8 o'clock here in the morning, and
right.
I woke up and the first thing I saw is I was seeing if the rain had stopped for the cricket match. So that's good news. Cricket is on again. I'd like to thank Dr. Kher, and I'd like to thank the organizers for giving me an opportunity to present our research work at this conference. And in a way, I would say this is one of the very few beneficial side effects of COVID-19, that we can really get virtual and take part in these conferences, which would otherwise not be possible. So I'm really happy to present my research work here. So I've been working on grapes for more than 15 years now, and I started doing research on grapes from my postdoc, and then I was very fortunate to continue my research program in the same areas. So I'm going to talk about the advances we've made with in the area of precision breeding for improving traits in grape vines. And okay, let me just move this. All right, that should be better. Okay, so grape is the most valuable fruit crop in the world. Of course, in the United States, it accounts for more than 30% of the value added by most fruit crops, basically coming from the value of wine. And it creates a number of jobs in the United States every year. More than 1 million jobs, has a huge contribution to the national economy. And I was looking at the numbers on grapes in India. So it's a big crop in India too. So we have grapes on more than 300,000 acres. Most of it is table grapes, but again, the wine grape industry is picking up really fast in India. We have a decent sized grape industry in Maryland. We have around 1,000 acres of grapes, and it contributes around $47 million to the economy every year. And so there is a great thrust on the expansion of vineyards and the expansion of grape production in Maryland. Another interesting fact is grape is grown in every single state in the United States. So it's a really important crop because in addition to just growing the crop, it stimulates the local economy. When you have a vineyard, when you have a winery, you have a lot of people that stop in that place or in that town that has the winery. They taste the wine, they spend the money, and they're just getting tax dollars to the local economy. So if you look at the limitations of conventional breeding for grapevine improvement, there have been certain breeding programs in the world. Most of these breeding programs have been focused towards the development of new table grape varieties. So table grapes are the grapes that are seedless and the grapes that you eat in the market, that you get in the grocery store. But with the wine grapes that contribute to more than 70% of the acreage in the world, we are still using the very ancient varieties. So Pinot Noir, for example, Pinot Noir is a very famous wine variety. And Pinot Noir was discovered by the Romans 2000 years ago. And we are still using those same clones that were discovered 2000 years ago. We are still using those clones by asexual propagation. And one of the reasons why we are using these varieties is they have extremely high enological characteristics. They make excellent quality wine. Now, if you look at the grape genome, the grape genome is extremely heterozygous. And so this is where traditional breeding becomes a problem, because if you're using the conventional breeding approaches where you take two parents and you make an F1 hybrid, the hybrid is basically of intermediate quality. So you might add certain traits, but it comes at the expense of disrupting other existing high quality traits. And so that's a problem, because when you look at the consumer exactly wants their wine to be the same quality as the parent, okay, which is why these ancient varieties are so valuable to the industry. And then there are certain incompatibility barrier between wider species. So, for example, the traditional bunch grapes, they have 38 chromosomes. The muscadine grapes, which is another species of grapes, which is highly disease resistant, highly drought tolerant, it has 40 chromosomes, and so basically you can't breed between bunch grapes and muscadine grapes. So you have certain problems with conventional breeding of grape wine, which is why there has been very limited advances that have been made for the development of new grape wine varieties. Like I said, we still use those very ancient varieties. So this is where precision breeding comes into play, and this is the only time I'm going to read my slide word to word, because this is a definition that we have coined for precision breeding. And so precision breeding is an approach to plant genetic improvement that transfers only specific traits among sexually compatible species via the relatively stable mitotic cell division pathway in order to avoid the significant genetic disruption imposed upon conventional breeding by meiosis. And so what we are doing here now is we are using the cell culture pathway or the tissue culture pathway. We are using the mitotic cell division pathway to insert traits of interest. And that way what we do is we keep the existing characteristics of any cultivar intact, but now we can add new traits in there. So that's a big advantage of precision breeding, because you're not getting that disruption that is imposed by conventional breeding. Okay. So what has made precision breeding of grape wine possible? So there's been a lot of work that has gone into making precision breeding now at the edge of commercialization. So of course in 2008, the grapevine genome was completely sequenced. And so now, we have the sequence of the grape genome. And so we can basically mine this genome for the traits of interest. Okay. We've developed an excellent grape cell culture and regeneration system. So the tissue culture system for grapes is really well developed, whether it's micropropagation or whether it's organogenesis or somatic embryogenesis. So this gives us an avenue to insert these traits of interest. Okay. And then again, we have excellent genetic engineering systems. So we have really good gene insertion systems to insert traits of interest, and then develop a large population that we can then screen for our trait of interest. Okay. So we have these three factors in place. And once you have these three factors in place for any commercial crop for that matter, then you can really advance the technology of precision breeding. Okay. So one of the questions that comes to your mind is, okay, how is precision breeding different from transgenic breeding? Okay, so how is it different from transgenics or GMOs or genetically engineered crop? Okay. And so if you look at the big difference or the major difference is the source of the DNA that is inserted in the plants. So where does your DNA, or where do your pieces of DNA that you insert in the plants to add these improved traits, where does it come from? Okay. So you can find different definitions for how these sequences come up. So for example, you have a cisgenic sequence. Okay, so a cisgenic sequence is basically a native unaltered sequence. So now you have the grape genome, so you can pick up the promoter, your coding sequence, your terminator. You can pick up the whole sequence from the genome, and you can insert it in a sexually compatible species. Okay, or you can use an intergenic sequence, where say you have a promoter from chromosome 2, a coding sequence from chromosome 19, or terminators from somewhere else. It's all coming from Grape. So again, it's all coming from either Grape or a sexually compatible species. And so it fits the definition of precision breeding. Okay. And then you compare it to a transgenic sequence where your promoter is coming from a virus, your coding sequence is coming from a bacteria, or it's coming from an animal, your terminator is coming from a bacteria. And so what you're doing here is you're getting your sequences across these plants and animal kingdoms, and you're stitching them together and inserting them in plants, which is why there is so much opposition to GMOs, right? Because you're carrying out horizontal transfer of DNA pieces. Okay, and so this is where precision breeding becomes really different from transgenic breeding, is where your pieces of DNA that you're inserting in plants for improving your traits are coming from sexually compatible species. Okay, so how have we come to the stage where we have optimized this technology? Okay, so like I said, we have really good cell culture systems. If you look at the genetic diversity for grapes in the world, there are more than 5,000 to 7,000 varieties of grapes. So there is a huge amount of genetic diversity in grapes, and it becomes important to customize your cell culture systems. It becomes important to optimize your cell culture systems for specific varieties. Okay, the same thing goes for gene insertion. All this diversity, you have to optimize protocols for efficient gene insertion systems. And then, of course, you have the grape genome that you can identify different elements. You can identify different candidate gene sequences, and then you can just carry out the functional testing on a massive scale. Because once you have a good engineering system, then it just becomes a matter of assembling your pieces of DNA together into your genetic cassettes, and then carrying out functional analysis. Okay, and so this functional analysis is, it can be overexpression or it can be knockouts. But what this helps you do is it basically helps you know the function of the specific promoter elements or the function of the specific genes in plants in grape, for example, here. And that helps you use these sequences for trait improvement. Okay, so this is an example of a culture system for grape. So this is where we use leaf explants. If you look at the top picture, and you put these explants in the dark, followed by a light cycle, and then they divide into these sectors of embryogenic and non-embryogenic callus. And then once you put them on development medium, you get these grapevine embryos that can be then used as target sequences, right, for inserting genes. Now we've seen that among all the varieties of grapes that we've tested, the leaf explants, they only work well for the seedless grapes. So if you test the seeded grapes, which are all the wine varieties, these leaf explants, you can't get embryos from leaves. And so you have to go for the floral explants. Okay, so we use these immature inflorescences, and same process, put them in the dark for 7 weeks, followed by light exposure. And then they give you these embryos, very high quality embryos that you can use for gene insertion. Okay, so we have tested more than 75, 80 different varieties of grapes, and we've developed around 8 to 10 media combinations for these varieties. And all the varieties that we test, they work on one of these media combinations. So a really efficient system for regeneration, and a really efficient system for insertion of traits of interest. Okay, so then we also have a good gene insertion system, and this is an example where we would use the green fluorescent protein as a reporter gene. And so what you do is you take your embryo explants, you co-cultivate them with your agrobacterium culture that has your construct with the genes of interest. You co-cultivate it for a period of 2 days, around 2 to 3 days in the dark. Then you wash your explants with antibiotics to get rid of the agrobacterium. And then it goes through a cell culture process where you induce callus. This callus is now transgenic in nature. Then you transfer to development medium, it gives you embryos. And now you can see these somatic embryos, they stably express the green fluorescent protein. So based on green fluorescence, you can identify your independent lines and you germinate them. And now you get your modified plants, right? Now, if you look at these modified plants, the expression systems are very efficient. So they are, depending on the kind of promoter that you're using, these expression systems. So in this case, if we use a 35S promoter, which is a constitutive promoter, you can see here we are expressing these at the cell culture stage. This is in your somatic embryos. Then you go through a callousing phase. You go through the process of repetitive embryogenesis, and you get these transgenic somatic embryos that express green fluorescent protein. And when you germinate this plant, you can see this is a mature grapevine that is in the greenhouse. It's in a pot. It's flowering and fruiting, and you can see the expression is very efficient. So you can see the expression in the leaves, in the flowers. You can see the expression in the reproductive structures, and you can also see the expression in the fruit. Okay, so your systems are very efficient. Okay, so now you have your two pieces of the puzzle in place, where you have a good tissue culture system, you have a good gene insertion system. Okay, so now the next step is where you are looking for your elements and your genes of interest. Okay, so if you look at all the crops, the first generation of GMOs in the United States, and the top three are corn, soybean, and canola. And these three crops, they account for more than 93% of the acreage in the United States. So genetically modified crops are grown on a huge area in the US. And by far, the number one trait that they use is herbicide resistance, because it's just easy to grow these crops. Okay, so if you look at how these first generation GM crops have been developed, your reporter gene has come from jellyfish or E. coli. The promoters are coming from different places. Your selectable marker is coming from E. coli. Your border sequences are coming from agrobacterium. That's how you're enabling the transfer of the tDNA. And so now our goal here is we have to replace these pieces of DNA. We have to make our genetic cassette completely derived from grapevine elements. Okay, so it has to come completely from grapes. Okay, so in order to do that, we started testing different candidate genes and different promoters from grapes. Okay, so one of the candidate genes we selected for the reporter gene was a MIP transcription factor that is involved in the anthocyanin biosynthetic pathway. And so if you look at the evolution of grapes, historically, if you look at the evolution of grapes, all grapes were red or black to begin with. Okay, and then over a period of evolution, somewhere there was a transposable element that got inserted in the promoter region of the MIP transcription factors, and that's what caused a mutation. And then you had the evolution of the white grapes. Okay, so the whites evolved from the red because of this mutation. But the interesting part of this mutation is it's not a very stable mutation, because it's a retrotransposable element, and so it keeps kind of jumping in and out. And so every now and then in the vineyard, we will see there is a white grape variety and it turns red in color. And so there might be a wine that, one single branch is having like a bunch that is different from the others. Okay, and so that's because this retrotransposon is kind of unstable. Okay, and so if you look at where is the MIP transcription factor, it's right here in the biosynthetic pathway. So it is the final step that is involved in the production of anthocyanin. And that's one of the genes we wanted to test, is if we could use it as a reporter gene. Okay, we could substitute it as a reporter gene instead of, say, your green fluorescent protein, your GFP or your GUS. Okay, and it's a visual indicator, easily detectable. And the most important part is it allows you to track your modified events in a non-destructible manner. Okay, plus you don't need any expensive equipment. With GFP, you need like a $20,000-$30,000 microscope to test your modified events. And with anthocyanin, you can just see this with the naked eye, right? So it's really easy to detect. So what we did is we isolated this MIP transcription factor. We expressed it under a constitutive promoter. And you can see here, we get really good transient expression after 3 days. In around 3 to 4 weeks, you see callus formation in here. And then again, you transfer these calli to embryo development medium. you get these pink or these red color embryos through the development of process of repetitive embryogenesis. And then you go and germinate these embryos, and you can see these embryos are completely expressing the anthocyanin. Okay, so you can see that now you can replace the green fluorescent protein or the GUS gene or any other reporter gene with a plant-derived gene. Okay, so we took these to the greenhouse. You can see here these plants in the middle are our controls, and all the other ones are expressing anthocyanin at these are all independent lines. So depending on where your gene is getting inserted, how many copies of the genes are getting inserted, you can see that they express different levels of anthocyanin. Okay, and this was work done by my graduate student who got his PhD and is doing his postdoc now. So you can see here the recovery of modified events is kind of comparable. If you compare it with GUS and GFP, you can see there is no decrease in the efficiency of the recovery of modified events. So one of the things we want to see is we do not want to compromise the efficiency of our system, right? If we are using an endogenous reporter gene. And so that's what we see here is with both varieties, we use two varieties here, Thompson seedless and Bronx seedless. You can see that the recovery is pretty good with both these systems. So we know the system works really well. And now we can replace a transgenic reporter gene with an endogenous or a precision bred reporter gene system. Okay. So this is a really good system, but there is a problem here. Okay, because when you're producing your plants commercially, you don't want your plants, all red looking in the field, because we did some studies where we could see that expression of really high levels of anthocyanin could actually interfere with the normal growth and development of the plant. Okay, so what we wanted to do is we wanted to switch our genetic element with a grape genetic element to where it would express only at the tissue culture stage. Because if you look at the function of the reporter gene, it is to help you identify your modified events, right? Once you get a modified event and once you get a plant, that basically that reporter gene is not needed anymore. Okay, so what we did is we used a seed-specific promoter from Grape. We used a 2S albumin promoter, and we hooked it to our anthocyanin gene. And you can see here that you get good expression, you get good transient expression, good callus expression. But once your embryos germinate and you get plants, that expression is stopped. Okay, so it is supposed to express only at the cell culture stage. And once you get mature plants, those mature plants are basically normal plants. So you can see here, this is our control here, which is a non-modified plant. This one here is modified with the anthocyanin, but it is expressing it under a different genetic element. And this is expressing anthocyanin under a 35S promoter. Okay, so you can see here, this is totally red, like it's expected. This one is working like we thought it would work, where it is not expressing anthocyanin at the whole plant level. And of course, this is our control plant. Okay, so now what we've done is we have switched our promoter element from a 35S, which is virus-derived, to a grape-derived genetic element. And we have our reporter gene that is anthocyanin. So we have almost reduced, we have completely switched one side of our genetic cassette from being transgenic to being cisgenic. Okay. All right, so we also identified several promoters from grapes that were giving us comparable expression to the viral promoters. And so two of these promoters, we saw the ubiquitin, the 7-2, 7-5. The expression is not as strong as a 35S promoter, but still we could get a very high level of expression to where we could use them for functional analysis. Okay, so we use these two promoters in all our genetic cassettes that we currently make for testing our genes of interest. And we also used an acetolactase synthase gene to replace our NPT2 marker. Okay, so the NPT2 neomycin phosphotransferase, it comes from E. coli. So it's a transgenic marker. And so we use the acetolactase synthase, which basically, if you look at the function of the ALS gene, it is responsible for the production of amino acids. And so the ALS is a target for a lot of herbicides, like the sulfonylurea herbicides, for example. It can, that's what they target, and they inhibit the ALS gene, and that's what kills the plant basically when you spray the herbicide. But if you make these selective mutations, these point mutations in the ALS gene, then certain point mutations can give you herbicide resistance. And so we made these point mutations and we tested it in tobacco. And so one specific point mutation, when you insert the gene in tobacco under a 35S promoter, you can see that it gives you pretty good resistance to this herbicide when you add it in the tissue culture media. And so this is what we're doing right now in grapes, where we are testing how does this marker actually work in the grape system. Okay, so potentially you can use ALS as a marker gene instead of a transgenic marker. Okay, so then of course your final goal is to look for those functional genes that are involved in either biotic stress or abiotic stress. Okay, and so if you look at the grape genome, there are a number of proteins, the PR proteins that are produced in response to pathogen attack. Okay, or number of proteins produced in response to abiotic stress. And some of these proteins can give you functional resistance. So these proteins, if you do the comparison between the sexually compatible species, so there are a lot of wild species that are highly disease resistant. And so you see significant differences between the sequences from these wild species and your cultivated grape wine. Okay, and so now what you can do is you can isolate the sequences from these sexually compatible species, which you couldn't do otherwise using traditional breeding. And now you can insert them in grape wine to see if you can get disease resistance. Okay, so that's one of the candidates that we used. We used a Vitis vinifera thaumatin-like protein, and thaumatin is actually it's similar to miraculin, where it is a sweet protein. It imparts a sweet taste. And there have been a lot of studies done in thaumatin, where they looked at the expression levels in grape berries, and they noticed that as the expression level of thaumatin increases in grape berries, the resistance to powdery mildew also increases. So this was study done in the 90s, and they could pinpoint that the VVTL1 was one of the genes responsible for disease resistance, for powdery mildew resistance. And so we took this gene, we again used our gene cassettes, inserted it in grapes, and we developed this large independent population of grapes that we tested in the greenhouse. And what we do is now testing for powdery mildew can be tricky because powdery mildew is an obligate parasite. And so you need a live inoculum to test the plants. So what we did is we just maintained this greenhouse where we wouldn't spray our greenhouse. And if you don't spray for grapes, then it's very easy to get diseases. It attracts a lot of pathogens. And so we had a very high inoculum of powdery mildew in here. And out of these 70 or 80 lines that we had, we could identify around 3 lines that were giving us certain resistance. So you can see here, this is a precision bred line here compared to your control that is giving you some resistance. And when I talk about resistance, it's a very relative term. So what we were seeing is a delay in symptom development by around 7 to 10 days, compared to the control. So if your control was completely covered with mildew by say day 4, then you would see those same results maybe around day 13 or day 14, okay, in your precision bred line. And so if you think about it, on an individual scale, it's not a big difference, right? I mean, it just gives you it gives you a resistance or a delay in symptom development by say 10 days or 2 weeks. But if you look at it in the context of grape management, so just for powdery mildew, grapes are sprayed around 22 to 25 times a year, for powdery mildew control, and that doesn't include the sulfur fungicides. The sulfur is considered organic, so if you take sulfur into account, you can go up to 30 times a year for spraying powdery mildew. Now, if you can delay resistance by 2 weeks, then you can cut your fungicide use by 50%. Okay, so that is dramatically reducing the amount of fungicides that you're using for spraying, just by delaying or inducing a resistance of 10 to 14 days. Okay, so that's where this becomes really important, because it's not a silver bullet. You're not getting a plant that is immune to powdery mildew, that it's not going to get powdery mildew. it is showing the resistance, and this resistance is helping you just improve the management of this crop by using less fungicides. Okay, so we took this out in the field. We got a field permit, a USDA field permit, because we had some transgenic elements in those plants. And in the field, it's interesting. In Florida, this is work I did in Florida. It gets really hot outside. And so once the temperatures cross 90 degrees Fahrenheit, which is around, I would say 35 Celsius, it is very hard to get powdery mildew in the field. But we were getting other pathogens. So we were seeing a lot of black rot in the field. And so we could see some resistance to black rot in the field. And so we could tell that the VVTL1 it's a good candidate because it's giving us broad spectrum resistance. It's giving us good results against mildew, which is by far the number one pathogen for grapes. But it is also giving us some resistance against black rot. So if you look at the whole system, you have your one side of the cassette. You have your promoter and your reporter gene and your marker gene coming from grape. You have some candidate genes coming from grapes, but your border sequences that you use, that the agrobacterium identifies for making the nick and then inserting it inside the nucleus, these border sequences you still use are bacterial in origin. Okay, and so if you truly wanted a precision bred cassette, you would have to look for those border sequences that would be homologous to the agrobacterium border sequence. Okay, and so we scanned the grape genome for those border sequences, and we found certain sequences that were homologous to these border sequences for agrobacterium. Okay, and so we replaced the agro border sequences with the grape borders, and we again tested it using GFP. And you can see here, this is your agrobacterium border sequence, extremely efficient, and kind of makes sense because agro has, evolved to infect plant species. But you can see here that the grape border sequences also gives you some amount of gene expression. So you can see here that they are, the bacteria is recognizing these borders. It is causing the cleavage of those border sequences and transferring the tDNA into the plant cells. Okay. So of course, the system needs to be refined because you can see a big difference in the expression, right, between the agro borders and the plant borders. But at least we know that these sequences, they can be used as candidates for our gene cassettes. Okay, so if you look at the bigger picture, where your pieces of DNA were coming from they were transgenic in origin. Now you're getting your pieces of DNA from grape itself, and now you have your precision bred cassette, where if you look at this implementation, if you were to commercialize this system, you have a good cell culture transformation system, you have a grape-based reporter and a promoter system, you have the ALS system, and of course you have the functional system, right? The functional candidate genes. So now you can completely derive your pieces of DNA from the grape genome or from sexually compatible species, and you can insert them for improving specific traits. Okay, so in our case, we are mainly looking at disease resistance. Now, what we have done is we have taken this one step further, where we're looking at genome editing. Okay, because past 8-9 years, CRISPR-Cas9, talons, other enzymes, zinc finger nucleases, they've been used for carrying out specific edits in plants. And so in this case, we are, if you look at CRISPR-Cas9, our system or our pipeline that we developed for precision breeding, it can be implemented for genome editing. Okay, because now you have the same pipeline, but instead of doing overexpression or instead of doing knockouts, you are using CRISPR-Cas9 and you're editing. Okay, so what we did is to do a proof of concept. We wanted to see how does CRISPR work in grape. So what we did is we used the PDS1 gene, the phytene desaturase gene, which is responsible for carotenoid biosynthesis, and we inserted it in a vector, did the transformation, same process, got our embryo lines, and once these embryos germinate, you can see here that they are completely devoid of chlorophyll, because PDS is one of the key genes in the chlorophyll biosynthetic pathway. Okay, and so if you compare it to a control embryo, which is normal looking, you can see the PDS1 here is completely bleached. Okay, and so you germinate the embryo, you get the plants. You can see these plants here they are barely surviving, right? Compared to a normal control. And if you look at the leaves, so this is your control leaf here. This is a complete knockout. So this is where CRISPR has worked really well. And this is I would say it's a leaky knockout, right? Where you can see that the PDS1 has been knocked out, but it is still making some chlorophyll. So maybe it's not all copies have been knocked out. Okay, it's a monoallelic or it's not biallelic. Okay, so we know that CRISPR works really well in grapes. Another trait that we used is for berry color, and this is what I mentioned before, is where you can see when the transposable element in the MIP transcription factor, it jumps in and out. You can see the change in color of the berries. So you can see here, this is a wine that has a normal gene, no mutation. And then that transposable element jumps in there, and all of a sudden you see this thing, you see a white cluster in there, right? And so we know that, if you can edit these transposable, if you can edit the MIP-A1 gene, then you can get different versions of the same variety, okay? Now, if you have a red grape variety and you want a white version using conventional breeding, it takes you a really long time because you have to use backcrossing, right? And so once you have grape seedlings, they take at least 2 to 3 years to flower. And so you have to add that into your backcrossing equation, and it takes a really long time. But in this case, now you can take a red grape variety and you can target the MIP-A1 gene using CRISPR-Cas9. And in a single shot, you can convert a red grape variety to a white grape variety, right? So what we did is we did a proof of concept again. So the plants where we were overexpressing the MIP-A1 gene to make them completely red in color, we thought, what if we knock out that gene? Do we revert back to the normal phenotype? So we produced embryos from those plants that were overexpressing MIP-A1. So this is what your normal embryos look like. This is what your MIP-A1 expressing embryos look like. And then we had a guide RNA target that targeted the MIP-A1, used agrobacterium. And you can see here, you get a mixture of edited and non-edited lines. So your edited lines, you can see here, they are reverting back to the normal phenotype. Your non-edited lines or the escapes are still giving you some kind of MIP-A1 expression. So this is what your control phenotype looks like, completely red in color. This is what your completely edited phenotype looks like. It's reverted back to normal. And then we get these intermediate phenotypes. So you can see here, in this case, the roots are red. In some cases, the roots are white, but the shoots are red. And so this tells us that, again, with CRISPR, it is not a very efficient system. You can get these intermediate phenotypes, just like we saw with the PDS1 too. Okay. But if you look at the commercial applications now, the MIP-A1 is a good target to where you can target this gene and you can get green or white versions of your red grape varieties. Okay, so that's a huge application in the table grape industry, because the industry currently, when they do conventional breeding, their annual budgets are between $12 to $15 million every year just for screening of their plant populations, because grapes are perennial. And so you need to put them in the field for a long time and screen them for at least 5 to 10 years before you can release varieties. So that increases their budget tremendously. Now, in this case, they have an avenue where in a single shot, they can change colors, right? So what are the commercial implications of precision breeding? They have huge implications, because the USDA issued a statement in 2018 where they said that they had a definition, and they said that there are certain, if you breed plants a certain way, say for example, deletions or insertions from sexually compatible plant relatives, these kind of plants will not be regulated as GMOs. And that makes a big difference, because it is easy to produce a GMO, but the process that goes into the deregulation and the testing process, that is an extremely cumbersome, time-consuming, and expensive process. So sometimes it can take 15 to 20 years of testing and deregulation before a product can come to the market. And so now USDA is saying that, okay, if you use precision breeding or if you use genome editing, then these varieties will be treated just like conventionally bred varieties. This makes a big difference to the industry, because now, it just saves them time and money. And so that's where precision breeding can be really applied to grape genetic improvement. So one of the things we are looking at, we just got a funded project here, is we're looking at powdery mildew resistance, where we are trying to use CRISPR to knock out these receptor genes for powdery mildew. Okay, they are known as the mildew locus O genes. And if we can knock out these receptor genes using CRISPR, then we can significantly improve powdery mildew resistance in grapes. Okay, there are other applications for CRISPR. So one of the applications is reduced acidity. If you can knock out the IDA gene, then you can just get an early crop, because your acid to sugar conversion ratio just happens much faster, and so your crop can come to the market earlier. So ultra early grape varieties are a big deal in the market in the US. Most of our grapes between January and May, June, they come from Chile. They're coming from outside the country, and so if a grower can have like an ultra early variety, then they can get a really high price for it. Okay, of course, mildew resistance is important, but then what we're also doing is, since grape is perennial and you don't do any breeding, we want to see if we can develop a transient expression system where you're inserting the gene, but or your CRISPR construct, you're carrying out the edits, but you're not allowing for integration of that, and so that's not classified as a GMO. So that's one of the things that we are still doing in the lab. I've had a lot of postdocs and visiting scientists from around the world. So we have a lot of people that come and want to learn about grape cell culture, grape transformation for CRISPR. And I do have a lot of graduate students too. So if you're interested, if we have a position, then you're definitely welcome to join us. Or if there are funding avenues in India that give you scholarships to come and learn these systems, then we can work with you. I'd finally like to acknowledge our collaborators. We work closely with the University of Florida. That's where I did my postdoc. And like I said, we have a lot of international collaborators from Canada, from China. We did a project with a university in Egypt where they were looking at drought tolerance. And all of our funding comes from USDA. We get some industry funding from the table grape companies. And of course, we are supported by our University Ag Experiment Station. So that's all I have. And I'll be happy to take any questions. Thank you.
Thank you very much, Professor Sadanand, sir. One ask how to join, sir, PhD program with you.
What was the question? You're breaking up there.
How to join, sir, PhD program with you, especially?
Okay. There are two ways. One is you need to give the standardized tests, of course, the GRE and the TOEFL and all those things, So you have, because all universities in the US look for those standardized test scores. But then, every now and then we have positions for graduate students from the grants that we get. And this is for all the other professors also. So if we have a position, we just advertise it. So, or, word of mouth, if we know someone, then we just tell them, hey, we have a position. And then you can just send your CV and, so there's a two-step. You need to get admitted by the graduate school and then by the department. And so if you get admitted, at both places, then you can join the PhD program.
Okay, what about postdocs, sir, with you?
I already have one postdoc right now. So, we just hired a postdoc for the grant that we got. But that being said, other opportunities are always coming up. So, if there is something available, we advertise it. So just keep an eye open.
Yeah, sure.
I was just going to mention that if there are, I'm sorry.
Yeah,
I was just going to say that if there are any short-term, I know there are, we have people coming from other countries, from Chile, from Canada, from Egypt, where they have these, they are sponsored by their governments. They have short-term training programs, they come for 6 months, they come for a year. And so if there are any, I don't know if DBT or anyone else has these kind of, scholarship programs, in India where they can send researchers for short-term training,
Yes, sir. Then one question is there, sir, whether in CRISPR-Cas technology, is there any use of tissue culture technology, or CRISPR-Cas technology can be used without tissue culture type of things?
That's a great question. So, there have been a lot of efforts being made to do CRISPR in planta, because I always tell my students, tissue culture is more of an art and less of a science. So of course you need to know the scientific background, but it has a lot of art. And that's one of the things why, a lot of researchers in India are such experts in plant tissue culture, because it's an art. There are efforts to develop CRISPR in planta. There was actually a paper that came out on Grape, I think like 2 years ago. But you can tell, from nothing ever comes out after that. So it's really hard to use CRISPR in whole plants, because really with tissue culture you're using the concept of totipotency. You're taking single cells, you're modifying them or editing them, and then you're getting whole plants. And so the products that you get from tissue culture, they're just more stable compared to what you would get with using CRISPR in planta.
Right, sir. Thank you very much, sir. It's really,
sure.
We all enjoyed. I think participants, including students, researchers, people who are waiting for PhD, who are waiting for postdoc also, right? Especially in the field of grapevine. It's eye-opener, sir, that this much of things are going on in the field of grapevine. Yes,
yeah,
sir.
I'm really happy to see
we
more than
learned
100
many
people,
things, sir, around 150 are there.
Yeah, that's great. Because
these
generally when
countries.
you're the last speaker, you struggle to have people, who are attending. So this is really great.
Really interesting to know about this technique, sir.
Sure.
This is technology, sir. Right, including development of grapevine derived reporter gene system, anthocyanins, biosynthetic pathway, development of grapevine derived selectable marker system, improving of disease resistance in grapevine, modifying berry color using CRISPR-Cas technology, and finally the commercial aspect of application of precision breeding and development of ultra early variety. Right, wonderfully we enjoyed, sir. Thank you very much, sir.
Sure.
And at this juncture, I would like to thank from my side to the university, GSFC University, the Chancellor, Vice Chancellors, Chancellor, sir, Deans, Head Department of Biotechnology, the convener, organizer, Dr. Mapatlalji, team of organizers, Dr. Sarojji Shekhawat, Samagani Ugam, then the GSBTM team, students, scholars. Thanks a lot for giving me this opportunity to share, to share this session. Thank you very much.
Good evening, everyone. Thank you, Dr. Shekhawat, for hosting the webinar, for moderating those afternoon sessions and taking out time for us. All right, my apologies for the disturbance in the background. Thank you, Dr. Sadanand, for a very exciting talk. I can see that there were a lot of requests to join your lab post your talk. You'll be probably bombarded with those requests later on as well. But thank you, sir, for joining us in our session. And thank you, Dr. Mahipal Shekhawat, for chairing all the sessions and moderating all the sessions and taking out time for this webinar. We are extremely grateful to you, sir. And now I would like to introduce the team of Gujarat State Biotechnology Mission. As you know that our international webinar has been financed by GSBTF. As I already said, that it's a Gujarat State Biotechnology Mission. It is constituted by Government of Gujarat under the aegis of Department of Science and Technology to promote, support and facilitate the development of biotech in the state. We have with us today Dr. Daksha Sakya, who's the manager at GSBTM. She carries out HR development activities in biotech sector in Gujarat state, and she also looks after biotech capacity building programs, internship and awareness events, and entrepreneurship development programs as GSBTM. I would ask her to, and I would request her to please, speak few words detailing about the GSBTM activities to our audience. Maybe like it would help them in case they have any proposals. So I invite you, Dr. Sakya.
Thank you for the nice introduction.
Dr.
Very good
Sakya,
evening.
I don't think you're audible. Probably something to do with your mic.
Am I audible?
Yeah, madam is audible.
Yes, audible. Yes.
Please. Yeah,
Okay.
very well.
Thank you so much for nice introduction. Very good evening to everyone. Actually, Gujarat State Biotechnology Mission is a state nodal agency which acts under the aegis of Science and Technology, Government of Gujarat, and it acts on the promotion, support, and the overall development of biotechnology in the state of Gujarat. So, GSBT has their own various activities. They do the support the research through their various scheme. For that they also support the research projects. They do fund the research projects. They do also run the various training programs like industrial training programs, entrepreneurship development programs. They do also organize summer internship programs as well. And they also, GSBTM also involved in the formation and the implementation of biotechnology policy in the state. So for the startup and incubation also, GSBTM has various activities. And GSBTM is always has supported all the development in the state for the biotechnology. When we have an enthusiastic biotechnologist in our state, and whenever they need our support in the any kind of if they need research support, we have a scheme for the researchers. We do have a schemes for the students, entrepreneurs. We do all the kind of schemes, research scheme GSBTM have. And if I would like to also speak for this event, and I would like to congratulate GSFC University for organizing such a wonderful event. And I think this is our first collaborative event in terms of this organization, because GSBTM has also recently funded some research projects also to the GSFC University. So I think and this event was very fruitful, and all the session, like it was a 21 sessions, and the expert was from all across the globe. So to invite them and to take in a single platform is a very hard work, and it takes so much efforts to bring all the speakers at the same platform. So I think this effort, GSFC University has done, it's like a very good job, and it's very courageous work of the GSFC University. So GSBTM is very grateful for organizing such events, and we are also looking forward for the such supportive and collaborative events. We will, I think, help people and regulators, biotechnologies, and experts to invite on a same platform and discuss the real issues and real subjects of the research. So thank you for the listening. Thank you. That's it.
Thank you so much, Dr. Sakya, for the encouraging words, as well as you rightly said, that this is just the beginning. And we're going to be having many more such collaborative projects with inputs from GSBTM. So thank you so much. Now I would like to invite Dr. Mafatlal Khair, the chief organizer of this event, and to conclude the ceremony with his vote of thanks. Dr. Khair, please.
Thank you, Dr. Shekhawat, for moderating this session. Thank you, dignitaries on the dais, all the attendees. Dear ladies and gentlemen, it is a matter of great privilege to be here for this august ceremony of international webinar series on plant tissue culture and biotechnology, current trends and future perspective, organized by School of Science, GSFC University, and sponsored by Gujarat State Biotechnological Mission, BST. It has been our pleasure to host all the participants of the webinar at GSFC University, India. We have been fortunate to have some of the eminent personalities working in the area of plant tissue culture and biotechnology. I'm sure that participants must have benefited by attending this conference. Although the world is hit by COVID pandemic, it still has opened new avenues and platforms where the entire world can interact with each other on platform like this as we are attending today. So we have tried to make the best use of this alternative to initiate the vibrant and meaningful discussion on current trends and future perspective in plant tissue culture and biotechnology. First and foremost, I would like to extend my gratitude to GSBTM BST for assisting and funding this international webinar series. Thank you, GSBTM team, for being with us, and we would like to extend our collaboration with you for future projects and webinar series too. I am extremely thankful to you, our Provost Sir, Professor Nikhil Javeri, Director Shri Ramesh Panchal, Campus Director Shri Atul Dholakia Sir, Professor Saurabh Shah, Dean, School of Technology, Associate Dean Dr. Santosh Kumar and Dr. Nishit Parikh, and entire GSFC University family and GSFC University management team for their constant support and motivation to conduct this extensive international webinar series. We are also extremely grateful to all the eminent national and international speakers and eminent and learned moderators who accepted our invitation to deliver their deliberation despite in short notice. Their presence has attracted participants across the globe. Their research work stimulated discussion and interest in attendees. We are thankful to all the participants for overwhelming responses and active participation in the program. We are thankful to you for keeping the session interactive. I am thankful to all my fellow mates and colleagues, especially entire biotech faculties, university IT team, and administrative team for all the possible support for successful organization of this event and help to generate wide publicity. At the end, I once again thanks everyone for making this event huge success. Thank you. Madam.
So we can leave now.
So we thank everybody towards the end for joining us today. Thank you. For the participants who are interested in recordings of all the sessions, all the recordings are available with the link shared. And you can also access the recording from our GSFC University website, and it would be posted there. The links are posted under the YouTube link, so you can access it from there. And I wish all the participants, I think, more meaningful interactions in the future as well. And I greet you, Sir Dekne sir. I wish you a good day ahead of you, since you're just starting your day there. We're looking forward to like ending the day here. And I thank you, ma'am, Sankhya ma'am, for joining us today. And thank you, Shekhawat sir. Thank you so much for hosting today's webinar. Good evening, everyone, and good day, sir. Greeting from team of GSFC University. Bye-bye, everyone.
Bye-bye.